Okay, so with our understanding of cells, now we're going to start understanding organisms. But before we do that, we have to focus on feeding and reproduction. And believe it or not, the kind of cell you have fundamentally determines how you're able to reproduce and how you're able to feed. Okay, the level of a cell can control at one level or determine what the entire organism is capable to do. Again, every organism has one or more cells, and so the cells are fundamentally important in building the organism, the structure, and what the organism's able to do, the function. And so let's go through that and focus on reproduction. Okay, so here's a bunch of different organisms. Some are single-celled, some are multicellular, and the kind of cell they have fundamentally determines how they're able to reproduce, whether it's an animal or a plant or a protist or a fungus or bacteria or you name it. So let's talk about reproduction. So when it comes to prokaryotes, reproduction is always asexual. If you remember our discussion about the biological species concept, this is one of the frustrating kind of difficulties with that idea, which is that if you are prokaryotic, you have prokaryotic cells, you can't do sex. Your reproduction is simply by duplicating yourself. Now, how this process plays out can be quite complicated, and we're not going to go into those details. Again, you will in other classes. Usually it's called fission. Sometimes it's called cloning. Sometimes it's called asexual reproduction. We're going to basically lump it all together. I'm going to summarize it as cloning because for most people that idea is pretty clear. Okay, fission means to divide into two. This is also called cellular fission or cellular kind of replication sometimes. Um, you know it as this process called mitosis, interphase, prophase, metaphase, anaphase, telophase, all that good stuff you've probably learned before IPMAT. If you're not a fan of it, you will be because you're going to learn tons about this in other classes most likely. But in asexual reproduction, your offspring, unless a mutation happens during the copying of DNA, your offspring is genetically identical to you. Hence, I like the word cloning because it's a pretty simple idea. Now, when we talk about cloning animals, which is something that a lot of biologists are trying to figure out how that's possible, um, that's more complicated here. What I'm talking about is one cell duplicates itself into an identical offspring cell. Okay, so this is asexual reproduction. So here we have some pictures, and so we have bacterial cells. This is our good friend in biology, the model organism E. coli, which we'll talk about coming up, that is basically dividing itself, and you don't see the DNA in here, but the DNA is also dividing as the cell divides. This process, if you're a prokaryote, is the only way you can reproduce. It doesn't mean eukaryotes can't do it either. So here's an amoeba, which is a protist. It has a eukaryotic cell. You can't see the nucleus, but you can see these dots in here, which are different organelles. And so eukaryotes can also divide by cloning, by asexual reproduction, by what's called mitosis. Okay. In fact, here's a multicellular animal. You can't see the cells, but you can kind of make out there's a layer of cells on the outside, kind of in the skin layer. And here it's budding off, kind of like a wart, and it will continue growing into a full organism, and then it breaks itself off. And this is a form of asexual reproduction. So if you're a prokaryote, this is your only way you can reproduce, but a lot of eukaryotes reproduce this way as well, even if you're multicellular. Okay, the key is if you're a prokaryote, this is the only option you have. Eukaryotes are going to have tons of options. If you're a prokaryote, the only way you reproduce is through mitosis. Okay, um, I do want to talk about something. It's the closest kind of bacteria can get to sex. Sex we'll talk about in a little bit, but basically means we're exchanging DNA. Two different organisms are exchanging DNA in some way. Um, and bacteria fundamentally can't do that as a sexual form of reproduction, but they can do something that goes by different names called gene transfer or lateral transfer or horizontal transfer or vertical transfer. There are subtle differences in what these terms mean, but for this class, we'll just call them lateral transfer. And basically, the way to think about this is that, let me see if I can get my marker, is that you have kind of one kind of organism and it has its DNA. Oh boy, this is going to be tough. Okay, and so that's one organism with its DNA. I'm going to change my pen color, if it will let me, to green. No, I shouldn't use green in case you're red-green colorblind. Let's do I'll do orange. And so here we have another organism, and it has its DNA. Okay. Um, and basically, in lateral transfer, is that a piece of this DNA might get moved into a neighboring cell and get incorporated in that DNA. Let's put an arrow just showing you which way it got transferred. Okay, let me get rid of this marker now because that's a little bit crazy. Okay, so in lateral transfer, what you're doing is you're basically having one cell steal DNA from a neighboring cell. And this tends to be pretty common in bacteria because, again, there's no nucleus to store that DNA. So the DNA is just wandering anywhere in that cell membrane, and it might kind of leak out of the cell a little bit, and a neighbor might pick it up. Or the neighbor might steal it. So again, it doesn't have to be stolen. It could be just kind of it gets lost and then incorporated. But that's gene transfer. And so they're not intending to share DNA. It just is kind of an accidental or random kind of process. And this can happen with eukaryotes, but because the DNA is surrounded by a nucleus and the cell is much bigger, the chance of it happening is much, much rarer. And so here is a phylogeny. Again, we'll learn what some of these words later on, but for now, here's bacteria. Here's the group of organisms with eukaryotic cells that are called the eukarya. Here are us over here, animals, very closely related to fungi. Here's other protists, and then over here are the plants. These are the bacterial groups with prokaryotic cells, and these arrows are showing you some ways in which, in the history of life, DNA has been transferred from one cell into another. And you can see in general bacteria participate in this a lot more than eukaryotes, but it has happened. A famous example is that there's a group of bacteria called the Chlamydiales, and there's a genus called Chlamydia, which in certain strains can cause a disease that's actually one of the most common sexually transmitted diseases on Earth. It's called chlamydia. Um, and chlamydia is basically, from a medical perspective, pretty difficult to treat with antibiotics, in large part because antibiotics have been invented usually by bacteria or fungi to kill bacteria. And if you're applying antibiotics to kill bacteria, well, you're like chlamydiales that ought to be able to be killed because it's bacteria. The issue is some of these strains have actually stolen animal DNA in their evolutionary history. And so when you have antibiotics that are basically trying to kill bacterial cells, bacterial DNA, chlamydiales have a little bit of animal DNA in them, which means that they're going to be less kind of responsive to normal antibiotics. And so that's a famous medically relevant example where gene transfer actually changes how you approach medicine. It's very common in lots of other bacteria, but there are some interesting groups. For example, land plants stole something from cyanobacteria as well, which is kind of cool. So this can happen. It's very rare to get eukaryotes doing this, but again, it has happened. You can imagine how complicated this was. We knew that this could happen up until the 80s, but we assumed it was incredibly, incredibly rare. But in the 1980s, we started having the ability to sequence DNA. And in the process, we started kind of doing molecular phylogenetics using DNA to figure out phylogenetics. And you can imagine how frustrating this is, which is that let's say that the yellow one is an animal cell and the red one is bacterial cell, and we sequence the yellow gene. Well, the animal has this gene, and this bacteria now has that gene. And if we only look at one gene and do phylogenetics based only on the DNA in that gene, we might find that this, based on the DNA, this bacterial cell is very closely related to this animal. And then you might find that here's a group of bacteria that branches off of the animals. And that's clearly wrong. This is a special case where it is a homologous gene, but the ancestor of these two organisms, this animal and the bacteria, which goes way back here to the origin of life, the basal ancestor, did not have that gene. The gene evolved much later on, but in lateral transfer it got moved around. And so when we started doing molecular phylogenetics, we quickly realized, oh my gosh, transfer makes figuring out these relationships really, really frustrating, really complicated, and you often can get the wrong answer, which is why today we use many genes because the chance all of those genes will have been transferred is very rare. It can happen. It can sometimes steal enormous amounts of DNA, but it's very, very rare. So this is a really messy thing when we started to figure out. And now that we're doing this regularly, it turns out this is actually pretty common, especially in bacteria, if you have prokaryotic cells. Okay, um, we've got 5 minutes on this module, so we can do this. We need to learn a few more concepts here. So important ideas that we're going to be using a lot this semester have to do with how DNA is configured in cells. And we're not going to go into detail what a chromosome is, but a chromosome is basically a bundle of DNA. And so here's a eukaryotic cell, here's the nucleus. And here we're visualizing these bundles of DNA called chromosomes. They kind of look like an X. It's two strands of DNA. One part you got from mom, one part you got from dad. And the differences in here can be really important. You'll learn about those as well in other classes. Just know they exist. They're kind of bundles of DNA typically in the nucleus if you're a eukaryote, but not in a nucleus. Think of this as a prokaryote, but get rid of the nucleus here. This picture is incorrect, but they can also have chromosomes where the DNA is kind of also unpaired, but not in a nucleus. So get rid of this nucleus. This drawing is incorrect. Another way we can configure this is these are three chromosomes and these are three paired chromosomes. And the terms we use for this are haploid and diploid. And think of this, the word here is ploidy, Ploidy. Ploidy refers to the characterization of the chromosomes. If the DNA is diploid, not diploid, but diploid is how we pronounce it. We abbreviate it 2N. What it means is that the DNA is paired up into paired chromosomes. Okay, kind of like these X's here. In the haploid state, it's half the ploidy. We abbreviate it 1N or just the letter N. It could be lowercase, it could be capital. You'll see both versions. And here the ploidy is halved. It is not uh, paired up. Okay, so in a haploid cell, the DNA is on its own. In a diploid cell, it's paired up. And typically the way this is going to work is if it's paired, you get one pair from mom, one pair from dad. Okay, in the haploid state, it's just one. So very important terms. So with that, we can talk about what eukaryotes do. And when it comes to reproduction, it gets more complicated. Um, I think I can get through this in 2 minutes. We might have to cut this module short and pick it up in the next. So when we talk about sexual reproduction, that's one of the new tricks. In fact, it's one of the synapomorphies that eukaryotes invented, was this ability to do sex or sexual reproduction, in which case there are two parents and producing each offspring, and in the process we're reshuffling the DNA to produce a new combination of the genes. So in this way, mom has her set of DNA, dad has his set of DNA. That DNA is going to basically get shuffled around. We'll talk about how in a second. It's called meiosis. And then they get recombined into a new combination, hence recombination, in a new offspring. And in this case, when we talked about evolution, each offspring genetically is going to be evolutionarily different from mom and dad because it's a new combination of mom and dad's genes. The technical term for sex, if you're a biologist, because the word sex can be used in many, many different ways by people who aren't biologists, is to use the word genetic recombination. Okay, so this is the technical phrase. We're basically taking DNA from two organisms and recombining them together. And this is basically what's possible if you're a eukaryote. You don't have to do sex. If you're a eukaryote, you can still do asexual cloning or mitosis. But if you're a eukaryote, this becomes possible. And for here, I'm just going to say that this is meiosis. So you can think of the distinction between mitosis and meiosis at an organismal level as the difference between asexual reproduction and sexual reproduction. Now I'm going to pause this. We'll finish this up on the next module.